Molecular basis of inheritance, chapter 5 biology, molecular basis of inheritance. In this chapter we are going to study about DNA, search of genetic material, RNA world, replication, transcription, genetic code, translation, regulation of gene expression, human genome project, and DNA fingerprinting. Okay, in the previous chapter, genetics chapter, you have learned that inheritance patterns and the genetic basis of such patterns. At the time of Mendel, the nature of those factors regulating the pattern of inheritance was not clear. Basically, in the previous chapter, we have learned about Gregor Mendel and his monohybrid dihybrid crosses and the segregation of factors and all those things. The nature of those factors at that time, the nature, like what are these factors, was not clear. Over the next 100 years, the nature of putative or putative genetic material was investigated, culminating in the realization that DNA, deoxyribonucleic acid, is a genetic material. Basically, this genetic material was investigated in the next 100 years after the time of Mendel, and later they found out that DNA, which is deoxyribonucleic acid, is the genetic material, at least for majority of organisms. For most of the organisms, DNA is the genetic material. But in class 11, you have learned that nucleic acids are polymers of nucleotides. Nucleic acids are polymers of nucleotides. Basically, they are saying nucleotides are the monomers. Nucleotide is nothing but a pentose sugar, phosphate group, and a nitrogen base. Deoxyribonucleic acid DNA and ribonucleic acid RNA are two types of nucleic acid found in living systems. Basically, deoxyribonucleic acid and ribonucleic acid RNA and DNA are two types of nucleic acids. Here they are not talking about genetic materials, they are talking about nucleic acids found in living systems. DNA acts as a genetic material in most of the organisms. In most of the organisms, DNA acts as the genetic material. This can be asked as a statement. RNA, though it also acts as a genetic material in some viruses. It only acts as a genetic material in case of some viruses. It is found in all living systems, in most of the living system, but it doesn't act as a genetic material. It only acts as genetic material, main genetic material, in some viruses. And it mostly functions as a messenger. In normal living systems, it only acts as a messenger. RNA has additional roles as well. It functions as an adapter, structural, and in some cases as a catalytic molecule. It acts as an adapter molecule, a structural molecule, and even a catalytic molecule. In class 11, you have learned that structures of nucleotides and the way these monomer units are linked to form nucleic acid polymers. In class 11, you have learned the structures of nucleotides, basically pentose sugar, nitrogen base, and phosphate group, and the way these monomer units, single nucleotides, are linked to form nucleic acid polymers. In this chapter, we are going to discuss the structure of DNA, its replication, the process of making RNA from DNA, transcription, the genetic code that determines the sequence of amino acids in proteins, the process of protein synthesis, translation, and elementary basis of their regulation, the determination of complete nucleotide sequence of human genome during last decade has set a new era of genomics. The determination of complete nucleotide sequence of human genome during last decade as set in new era of genomics. Okay. In the last section, the essentials of human genome sequencing and its consequences will also be. This is a topic that is going to be discussed at the last of this chapter. That is basically determination to complete nucleotide sequence of human genome. And this determination led to a new era of genomics in the last 10 years. Let us begin our discussion by first understanding the structure of the most interesting molecule, of course, in the living system, that is DNA. In subsequent sections, we will understand that why it is the most abundant genetic material and what is its relationship with RNA. We see that in most of the systems there is DNA, and only some of the living organisms contain, not living organisms, basically viruses contain RNA as the main genetic material. Viruses are acellular, so we cannot join them in the living section. The DNA. DNA is a long polymer of deoxyribonucleotides. Basically, deoxyribonucleotides are deoxypentose sugar, meaning a lack of a single oxygen from the OH group, and there is a phosphate and a nitrogen base. And DNA is basically made of deoxyribonucleotides, so it's called a polymer of deoxyribonucleic acid, nucleotides. The length of DNA is usually defined as number of nucleotides. Basically, when DNA is made of nucleotides, of course it is defined in the length of DNA can also be measured in the number of nucleotides that is forming the DNA as well. And normally we know that DNA is a helical structure, so there will be two nucleotides bonding here. This is a hydrogen bond, and because of such a reason we do not measure the length of DNA as like a single nucleotides, but mostly as a pair of nucleotide referred to as base pairs. Basically, nitrogenous bases are forming hydrogenous bonds, so we call it as nitrogen base pairs, or base pairs in short. And we measure the length of DNA in terms of number of nucleotides. This is also the characteristic of an organisms. This is also the characteristic of an organism. For example, a bacteriophage, a virus infecting bacteria known as 5174, has 5386 nucleotides. Okay, bacteriophage lambda has 48,502 base pairs. E. coli Escherichia coli has 4.6 into 10 power 6 base pairs. In viruses, we saw that there are around 5,386 nucleotides and 50,000 nucleotides, but as we came to E. coli or bacteria, prokaryotes, it directly jumped into the millions from thousands. And in humans, it is 3.3 billion base pairs into 10 power 9. This might be asked as the number of base pairs, as bacteriophage known as 5 into 174 or 5x 174 has approximately 5,400 nucleotides or 5,386 nucleotides. A bacteriophage lambda has 48,500 base pairs or 502 base pairs, and Escherichia coli has 4.6 into 10 power 6 base pairs or 4.6 million base pairs, and the haploid content of DNA is 3.3 into 10 power 9 base pairs. Basically, they are saying haploid, So in case of diploid, it will be 6.6 into 10 power 9 base pairs. Basically, because we know DNA is two strands, so one strand, all of the single strands will have 3.3 into 10 power 9. If both strands, if we consider diploid, and we will have 6.6 into 10 power 9. And they are defining it as it is a characteristics of an organisms, as we can say a bacteria or anything is round in shape or rod like or bacillus in shape. We can say an organism has this many amount of nucleotides or base pairs as its characteristic. Wait, okay, this is not both strands, as base pairs we will mention or consider for both strands itself. This is not a single strand, this is haploid content, meaning only half of the number of chromosomes considered. Let us discuss the structure of such a long polymer. We are going to discuss the structure of the long polymer. Structure of the polynucleotide chain. Let us recapitulate the chemical structure of polynucleotide chain. DNA or RNA. A nucleotide has three components: a nitrogenous base, a pentose sugar or a ribose sugar in case of RNA, and deoxyribose for DNA, and a phosphate group. Okay, they said the chemical structure of a nucleotide. Basically, nucleotide is made of monomers of, DNA is made of monomers of nucleotide, and a nucleotide is made of a pentose sugar, which can be ribose in RNA and deoxyribose in DNA, a nitrogenous base and a phosphate group. And there are two types of nitrogenous base: purines, adenine and guanine, and pyrimidines, cytosine, uracil and thymine. Basically, adenine and guanine purines are double ring structures. In the structure, if we see, it is a double ring structure, and pyrimidines, cytosine, uracil and thymine, they are single ring structures. In pyrimidines, we see there are three nitrogenous bases, but in case of DNA, we only see adenine, guanine, cytosine and thymine. In case of RNA, we do not see thymine. We see adenine, guanine, cytosine and uracil. As they are saying here, cytosine is common for both DNA and RNA, and thymine, cytosine is common for both DNA and RNA, and thymine is present in DNA. Thymine is present in DNA, uracil is present in RNA at the place of thymine. A nitrogenous base is linked to OH of 1 dash carbon pentose sugar through a N-glycosidic linkage to form a nucleoside. Basically, let's continue reading first. Such as adenosine or deoxyadenosine, guanosine or deoxyguanosine, cytidine or deoxycytidine, and uridine or deoxythymidine. Basically, they are saying a nitrogenous base is linked to the OH group of the carbon pentose sugar through a N-glycosidic form to form a nucleoside. Basically, when there is only a nitrogenous base and a pentose sugar, it is called a nucleoside. When there is also a phosphate group, it's called a nucleotide. So basically, when there is a linkage between nitrogenous base and carbon, it is of course a nucleoside, and it is a glucosidic bond linkage. These nucleosides can be adenosine or deoxyadenosine when there is lack of oxygen, guanosine or deoxyguanosine, cytidine or deoxycytidine, uridine or deoxythymidine. Deoxy case DNA, so thymidine. So basically here uracil can be uridine or deoxythymidine. When a phosphate group is linked to OH fifth carbon of a nucleoside through phosphodiester bond, a corresponding nucleotide or deoxynucleotide depending upon the type of sugar present is formed. Basically, the first bond is with the pentose sugar. Here, if we see the first bond, this is the pentose sugar, and here we have the nitrogenous base, and here there is a carbon, and there will be a phosphate group like this. This is a this is the pentose sugar, this is the nitrogenous base, this is the phosphate group. This phosphate and pentose linkage is called phosphodiester bond or linkage, and this linkage is called N-glucosidic linkage. That is what they are saying here. When a phosphate group is linked to an OH of the fifth carbon of a nucleoside through a phosphodiester linkage or a phosphoester linkage, a corresponding nucleotide or deoxynucleotide depending upon the sugar present is formed. Basically, formation of nucleotide. Two nucleotides are linked through 3' and 5' phosphodiester linkage to form a dinucleotide. More nucleotides can be joined in such a manner to form a polynucleotide chain. A polymer thus formed has one end a free phosphate. Basically, this is 5. The DNA we see it is called 5' to 3' or 3' to 5' because here in the linking of the pentose sugar, if we see pentose sugar, the phosphate group is attached to the fifth carbon or the fifth position here 5-C. So that is why where the phosphate group is free here at the end of the or the start of the DNA, here this phosphate group is completely free here, and in this case here if we see this is linked to the third carbon and it is OH group, hydroxy group is free here, so it is the 3' position. That is what they are saying. A polymer thus formed has at one end a free phosphate moiety at 5' end of the sugar, which is referred to as 5' end of polynucleotide chain. Yes, as I said before, similarly at the other end of the polymer, basically 5' is phosphate, free phosphate, 3' is free hydroxy group. Five end of the poly, and this free phosphate group is the 5' end position of the whole polynucleotide chain. The sugar has a free OH of the 3' carbon group, which is referred to as 3' end of the polynucleotide chain. The backbone of the polynucleotide chain is formed due to sugars and phosphates. Here if we see, the nitrogenous base is bonding with the second segment, but the actual polynucleotide chain's backbone is the phosphodiester bond. Basically, phosphate is linking with the this carbon as well as this carbon, so it is a phosphodiester bond. The nitrogenous base linked to sugar moiety project from the backbone. The nitrogenous base are getting projected like this from this main backbone. In RNA, every nucleotide residue has an additional OH group. As I said before, normally there will be, in here there is actually one more OH group. In case of, in the chain here we have one more OH group, but in this case there is, in case of RNA there will be an OH group, but in case of DNA there is no OH group. This will reduce the reactivity of the DNA. As an additional OH group present at 2' position in the ribose. 2' is this position only. That position is the 2' group. Where there is a OH group in case of RNA. Also, it is good to remember the positions OH in case of RNA. Also, in RNA, the uracil is found at the place of thymine. In case of thymine, we see uracil. 5-methyl uracil, another chemical name of thymine. Thymine can also be called as 5-methyl uracil. Methyl is a CH3 group. Presence of this methyl group will differentiate thymine and uracil. DNA as an acidic substance present in nucleus was first identified by Friedrich Miescher in 1869. He named it nuclein. First identified by Friedrich Miescher in 1869. The question may be who discovered DNA or found that DNA was a acidic substance in nucleus. It was Friedrich Miescher in 1869, and he named it nuclein. Who named DNA as nuclein and said that it was acidic? In which year? Friedrich Miescher in 1869. However, due to technical limitation in isolating such a long polymer intact, the elucidation of structure of DNA remained elusive for a long period of time. Basically, because it was such a long polymer, and because isolating such a long polymer intact, it was very hard to do, and it took very long amount of time to do it. It was only in 1953 they discovered it in 1869 itself that it was an acidic substance present in the nucleus, but it was only in 1953 that James Watson and Francis Crick, based on X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin, proposed a very simple but famous double helix model for the structure of DNA. Basically, at 1953, James Watson and Francis Crick, two people, based on the diffraction data given by or produced by two other people, Maurice Wilkins and Rosalind Franklin, based on these people's diffraction data, X-ray diffraction data, they proposed James Watson and Francis Crick proposed a very simple but famous double helix model for the structure of DNA. One of the hallmarks for their proposition was base pairing between the two strands of polynucleotide chains. Basically, base pairing between the two strands, basically hydrogen bonding between the two pairs. However, this proposition was also based on the observation of Erwin Chargaff that for a double-stranded DNA, the ratios between adenine, thymine, and guanine and cytosine are constant and equal ones. Basically, they proposed this DNA helix model, seeing the diffraction data produced by Rosalind Franklin and Maurice Wilkins, who James Watson and Francis Crick. They proposed the double helix model. Why they proposed it? First reason, wait, molecular basis of inheritance. Watson, James Watson and Francis Crick, right? I think so. James Watson and Francis Crick. Francis Crick, seeing / observing X-ray diffraction data given by Maurice Wilkins and Rosalind Franklin, and Rosalind Franklin proposed the double helix model, double helical structure of the DNA. Reasons for proposing, reasons why they proposed a double helix model specifically. One, seeing that the bases, nitrogenous bases, are getting paired via hydrogen bonding, which is adenine to thymine or uracil, and double bond here, double hydrogen bonding here, and guanine to cytosine with a triple hydrogen bond here. Seeing this, and two, seeing that they are getting paired, and seeing and observing Chargaff Chargaff's rule, which is given by Erwin Chargaff. Given that ratios of adenine, number of adenine by number of thymine or uracil is equal to the number of guanine. Wait. The ratios between adenine and yes, that one. Number of cytosine are equal and are also equal to 1. Constant are equal to 1. Basically, both are equal in number. Seeing this, they proposed the double helix model. The base pairing confers a very unique property to the polynucleotide chains. They are said to be complementary to each other, and therefore, if the sequence of bases is in one strand is known, then the sequence in the other strand can be predicted. Basically, because this base pairing is, they're saying this base pairing is a very unique property, because if the sequence on the first chain, which is there are base pairs, sequence is nothing but the base pairs, where it is adenine, guanine, cytosine, then when we can easily say what the second chain will be, because we know that adenine binds to thymine or uracil in case of RNA, and guanine binds to cytosine binds to guanine. So the second sequence we found out, it is TCG for AGC. This is what they're saying here. Because of this unique base pairing ability of the DNA, with one strand itself we can easily find the other strand's sequence as well. Also, if each strand from a DNA, let us call it as parental DNA, acts as a template for synthesis of a new strand, the two double-stranded DNA, let us call them as daughter DNA, thus produced, will be identical to the parental DNA molecule. Because of this, the genetic implications of structures of the DNA become very clear. Basically, they are saying about DNA synthesis, or which we will see later in this chapter. If each strand from a DNA acts as a template for synthesis of a new strand, the two double-stranded DNA, the daughter DNA produced, would be identical to the parental DNA. Basically, they are saying it can produce itself, like its replication will be same as the parent, and the daughter will be the same in sequencing. That is double-stranded. The salient features of double helix structure of DNA are as follows. It is made up of two polynucleotide chains, where the backbone is constituted by sugar phosphate and bases project inside. The two chains have anti-parallel polarity. Okay, basically the first point is the main backbone is the phosphodiester bonding between the pentose sugar and the phosphate group, and the bases, the nitrogenous bases, project inside the DNA. The two chains, the second point, have anti-parallel polarity. It means if one chain has a polarity 5' to 3', the other has 3' to 5'. Basically, 5' as I wrote before, 5' is the free phosphate group and 3' is the free hydroxy group. When the first chain is here, there is a free phosphate group, which is 5'. Here there is a free OH group. And the polarity, the second strand, it will be from here. In this case, here there will be a free OH group, hydroxy group, so it will be 3', and here it will be a free phosphate group, which will be 5'. This is the anti-polarity. The bases in true strands are paired through hydrogen bonding, H hydrogen bonds forming base pairs. As I said before, adenines form two hydrogen bonds with thymine from opposite strand and vice versa. Similarly, guanine is bonded with cytosine with three hydrogen bonding. As a result, always a purine comes opposite to a pyrimidine. We know that adenine and guanine are purines, whereas thymine, uracil, and cytosine are pyrimidines. And we see adenine is bonding with thymine and guanine is bonding with cytosine. Purine is bonding with pyrimidine. This generates approximately uniform distance between the two strands of the helix, because it's always either purine and pyrimidine bonding together, purine and pyrimidine bonding together, or pyrimidine and purine bonding together. All the bond lengths will be same, approximately same bond length. So the distance between the helix will also be same, and it will be uniform throughout the all of the helix. The two chains are coiled in a right-handed fashion. They will be coiled towards the right side. The pitch of the helix is 3.4 nanometer or 34 Armstrong. A nanometer is 1 billionth of a meter, that is 10 power minus 9 meter. And there are roughly 10 base pairs in a turn. Basically, they are described here that the two chains are coiled in a right-handed fashion. Important to remember, a right-handed fashion. And the pitch of the helix, which is 34 Armstrong or 3.4 nanometer, and basically they are saying there is 10 base pairs in these 34 Armstrong. Basically, it is turned like this. So in such a case, this is a single pitch which is around 34 nanometer, and in this pitch there are approximately 10 base pairs, and if there are 10 base pairs in 3.4 nanometer or 34 Armstrong, 10 base pairs, then we can find the distance between each base pair is 34 by 10, which is 3.4 Armstrong is the distance between each base pairs in a turn or in this pitch. And this is a double stranded polynucleotide chain. This is the glycosidic bonding, and here if we see, the OH is free here, so 3' to 5', here it is phosphate is free here, so 3' to 5', and here it is phosphate group is here free here, so 5' to 3', here it is a hydrogen group, which is hydroxy group, which is free. And as I showed here, it is the cytosine and guanine triple hydrogen bonding. Here guanine and cytosine triple hydrogen bonding, adenine and thymine double hydrogen bonding, adenine thymine double hydrogen bonding. Consequently, the distance between a base pair in a helix is approximately 3.4 Armstrong or 0.34 nanometer. DNA double helix, adenine guanine bonding, guanine cytosine bonding, sugar phosphate backbone. This blue color one. The plane of one base pair stacks over the other in the double helix. This, in addition to hydrogen bond, confers stability of the helical structure. The plane of one base pair stacks over the other in a double helix. Okay, this, in addition to hydrogen bonds, confers stability to the helical structure. Basically, they are saying stability of the helical structure because one of the base pairs, it is getting stacked on top of each other. So in such a case, it will provide stability. Compare the structures of purines and pyrimidines. Can you find out why the distance between the two polynucleotide chains in a DNA remains almost constant, as I said before? Purines are basically double circle structures which have a little larger wavelength, not bond length. But pyrimidines are single circle structure or single cycle structures, so they have a little less bond length. When these both are forming bonds of the hydrogen bonds, only these two can form hydrogen bonds. Because of such a reason, no matter which side if we take, there will be only purines and pyrimidines bonding. And if the bond length of purines is x and pyrimidines is y, there will always be x plus y itself is the bond length. No matter if it is y and x bonding in the left side here, this is a chain, and here x and y bonding, it will always be the total bond length will always be x plus y. So such a reason, the total distance between the strands will always remain constant. That is the answer. The proposition of a double helix structure of DNA and its simplicity in explaining a genetic implication became revolutionary. They are saying the proposition of this double helix structure of DNA proposed by James Watson and Francis Crick, seeing the diffraction data given by Morris Wilkins and Rosalind Franklin. Very soon, Francis Crick proposed a central dogma. Francis Crick is also the one who joined with James Watson to give the actual helix structure, and this Francis Crick is also the one who a little later or very soon after proposing the double helix gave the central dogma in molecular biology, which states that genetic information flows from DNA to RNA to protein. This is the central dogma, basically DNA self-replication, a semi-conservative nature of DNA, which we will see, and also the experiment for proving the self-conservative nature. And this DNA is self-replicating, and this DNA will transcribe to give mRNA, which is acting as a messenger, and mRNA will help in translation, which will create protein, and protein will control and regulate the body's needs and such, or body's physical expressions. Expression is not facial expression, but physical expression, physical expressing of the body, how the body is, how the growth or anything like that. In some viruses, the flow of information is in reverse direction, that is from RNA to DNA. Can you suggest a simple name to the process? I'm not too sure what we can name it, but basically they are saying in some organisms there is also reverse transcription. Wait, I just named it. Reverse transcription. Transcription is from DNA to RNA, and from RNA to DNA in some organisms it will happen, so we can call it as reverse transcription. Reverse transcription. Now let's see packaging of the DNA helix. If we see in a single cell, there is approximately 2-meter long length of material, DNA genetic material in a single cell. So it should be packed in a very, very minute cell. So we need importance in packing of DNA. Taken the distance between two consecutive base pairs as 3.4 Armstrong or 0.334 nanometer, if the length of the DNA double helix in a typical mammalian cell is calculated simply by multiplying the total number of base pair with distance between two consecutive base pairs, that is 6.6 into 10 power 9 base pair into 0.3 into 10 power minus 9 meter per base pair. it comes out to be approximately 2.2 meters. Basically, this 6.6 into 10 power base 9 base pairs or 6.6 billion base pairs are present not in the whole body of a person but in a single cell. As we see in Escherichia coli, there were approximately some million or 2 to 3 million base pairs that is in a single cell of Escherichia coli. The same way, in a mammalian cell, there are approximately, or a human cell, we can say, there are approximately 6.6 billion base pairs. And multiplying these base pairs with the distance between each base pair, which we calculated based on the pitch of the helix, which was 3.4 nanometer, we can say, and we know that in each turn or each pitch there is around 10 base pairs, so we can say the distance between each pair, if uniform, is around 3.4 Armstrong. And multiplying the number of base pairs times the distance, we will get it around 2.2 meter, a length that is far greater than the dimension of a typical nucleus. The nuclear material is not packed inside a cell, it is packed inside an even smaller nucleus. The dimension of the nucleus is approximately 10 power minus 6, which is in the millionth. Here we take a meter, which is a million times greater than the size of the dimension of the nucleus. How is such a long polymer packed in a cell? Let's see the core of a histone, the nucleosome first. This is the nucleosome structure. This is the ball-like structure is the histone octamer, and this is the DNA, which is wrapping around here around this histone protein. And this is the H1 histone. This is the histone octamer, and the whole thing is called nucleosome. These are the individual nucleosomes. This is an individual nucleosome. These are polymers or many nucleosomes. It looks like beads on a string. If the length of E. coli DNA is 1.36 mm, can you calculate the number of base pairs in E. coli? Basically, if the distance is the length is 1.36 into 10 power minus 3 meter, then by distance between each base pair, that is, I think, 0.34 into 10 power minus 9. So if we solve this, we will get the number of base pairs in E. coli, which was around a million, which we saw in the first page or so, the second page or so actually. In prokaryotes such as E. coli, though they do not have a defined nucleus, the DNA is not scattered throughout the cell. Even though in prokaryotic cell we know that there is no nuclear membrane and it is actually a nucleoid which is present. That's why we call it as a primitive karyote or a primitive nucleus. DNA being negatively charged, DNA is not scattered throughout the cell. Even though it is prokaryotes, it is not scattered throughout the cell. DNA being negatively charged, DNA is negatively charged because of the groups which is present in the nucleotide, which can be phosphate group. Because of such a reason, it will have a negative charge, and because it is negatively charged, it is held with some proteins that have a positive charge, positive and negative attraction, so it is held with positively charged proteins in a region termed as nucleoid. As I said before, in prokaryotes there is no nucleus, there is only a nucleoid. The DNA is nucleoid is organized in large loops held by these proteins, positively charged proteins. In eukaryotes, this organization is much more complex. There is a set of positively charged basic proteins called histones. A protein acquires charge depending on the abundance of amino acid residues with charged side chains. This can be important. In eukaryotes, the organization is much more complex. There is a set of positively charged basic proteins called histones. A protein acquired, they're saying how a protein acquires charge. The next information is a protein acquires charge depending upon the abundance of amino acid residues with charged side chains. Histones are rich in the basic amino acid residues lysine and arginine. Both the amino acid residues carry positive charges in their side chains. Basically, there will be a main chain and there will be a side chain, and determining a protein if it is having charge or not determines on the type of amino acid and a type of charge carried on their side chains. And histones are rich in basic amino acids residues of lysine and arginine. These lysine and arginine both carry positive charges in their side chain. Because of that, histones are positively charged basic proteins. Histones are organized to form a unit of 8 molecules called a histone octamer. Okay. Histone octamer is basically 8 histone proteins joined together. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome. A typical nucleosome contains around 200 base pairs of DNA helix. If it is around 200 base pairs, dividing it by a single pitch, a single pitch is around 3.4 nanometer, we will get the total number of turns possible in that DNA. By 10 actually, because there are 10 base pairs in each string, so 20 turns of the DNA. And these turns are not around the octamer but around the other structure. Nucleosome constitute the repeating unit of a structure in nucleus called chromatin. Nucleosomes constitute the repeating unit. Basically, chromatin, which is the DNA material, is actually made of monomeric repeating units called nucleosomes. Reverse statement is nucleosomes constitute the repeating unit of a structure in the nucleus called the chromatin. Thread-like stained colored bodies seen in nucleus. Thread-like stained or colored bodies seen in the nucleus. The nucleosomes, chromatin, are seen as beads on a string. The electron microscope picture, this is the structure. Structure when viewed under electron microscope. Theoretically, how many beads, such beads, nucleosomes, do you imagine are present in a mammalian cell? Basically, in a single mammalian cell, there are 6.6 into 10 power 9 base pairs. Then each nucleosome contains 200 base pairs. So 3.3 into 10 power minus 7. This is the number of nucleosomes which can approximately find in a single cell. The beads on a string structure in chromatin is packed to form chromatin fibers that are further coiled and condensed at metaphase stage. The beads on a string structure in chromatin is packed to form chromatin fibers that are basically this chromatin, we know that from the cell division cycles chapter. Chromatin is what condenses to form chromosomes. That is what they are saying here. In the metaphase stage of the cell division to form chromosomes, they get condensed, further coiled up to form chromosome. Packaging of chromatin at higher level requires set of proteins that collectively referred to as non-histone chromosomal proteins. Basically, this further coiling condensation of chromatin requires other proteins as well, and these proteins are referred to as non-histone chromosomal proteins. In typical nucleus, some region of chromatin are loosely packed and stains light are referred to as euchromatin. Basically, there is chromatin which we saw here, which was arranged like beads on a string. It can be loosely arranged like this and stains lightly, light stain, not much, like it cannot contain much stain, so it is light in color, light color stain, and this is referred to as euchromatin. This loosely packed is called euchromatin, and chromatin that is more densely packed and stains dark is called heterochromatin. This is heterochromatin. This is tightly packed, and due to the gaps we can say it stains very darkly or densely. Dark stain is called heterochromatin. This chromatin is more densely packed and stained dark is called as heterochromatin. Euchromatin is said to be transcriptionally active chromatin, whereas heterochromatin is inactive. Basically, they are saying euchromatin can transcribe to give rise to mRNA, whereas heterochromatin cannot transcribe to give mRNA. So it is inactive. Heterochromatin is inactive, euchromatin is active. So active chromatin is what we call as euchromatin. Now let's start with the search for genetic material. Friedrich Miescher already discovered nuclein, which is said to be acidic nucleic acid present inside the nucleus in 1858 itself. That is what they're saying. Even though discovery of nuclein by Friedrich Miescher and the proposition for principle of inheritance by Mendel were almost at the same time, both Mendel prescribing inheritance and Miescher finding out nuclein are at the same time. Like even Mendel proposed at around 1850s or 60s itself, but that the DNA acts as a genetic material took long to be discovered and proven. Even though the main information basis was still there, because people did not recognize Mendel's work and other such lags, we can say, the actual discovery of DNA and as a genetic material, and for it to be discovered and proved, it took very long time. By 1926, a quest to determine the mechanism for genetic inheritance had reached the molecular level. Previous discoveries by Gregor Mendel, Walter Sutton, Thomas Hunt Morgan about the genetic maps, and not genetic maps actually, it's about the linkage and recombination. Walter Sutton and Gregor Mendel both about the factor factors. Basically whatever Gregor Mendel did, Walter Sutton also independently researched and did the same thing. And numerous other scientists had narrowed the search to chromosomes located inside the nucleus of most cells. But the question of what molecule was actually the genetic material had not been answered. They understood that chromosomes were the one that were basically seeing all the research and all. They understood chromosomes were the one responsible for being the genetic material, but they didn't understand what molecule is actually the one that is responsible for the carrying of the genetic material and transfer. The first test we see is the transforming principle. In 1928, Frederick Griffith, in a series of experiments with Streptococcus pneumoniae, bacterium responsible for pneumonia, witnessed a miraculous transformation in bacteria. Frederick Griffith did an experiment with Streptococcus pneumoniae, a bacteria that is responsible for pneumonia. Pneumonia is basically a disease where in the alveoli a fluid gets filled and reduces the gas exchange rate. This is the pneumonia disease. And Frederick Griffith witnessed miraculous transformation in bacteria. During the course of his experiment, a living organism, bacteria, had changed in physical form. Let's see how that is. In Streptococcus pneumoniae, basically there are two types of Streptococcus pneumoniae. One is smooth strain, S strain. S is the smooth strain. This is due to presence of gelatinous layer. And there is R strain, rough strain. There is rough structure on the surface, rough like this, and here it is completely smooth due to gelatinous layer. It is rough inside, but it is smooth because of presence of gelatinous layer. And S strain, R strain types of bacteria. And when we inject S strain type of the Streptococcus pneumonia into the mice, a lab testing animal, a mice, the mice will die. For S strain bacteria, the mice will die. But for R strain bacteria, when we inject into the mice will live. Meaning S strain bacteria can kill the mice. The smooth strain will kill the mice. But R strain bacteria isn't strong enough to kill the mice. And let's continue reading. When Streptococcus pneumoniae, pneumonococcus bacteria are grown on a culture plate, some produce smooth, shiny colonies, S, while others produce rough colonies, R. This is because the S strain bacteria have a mucus polysaccharide coat, while the R strain do not. Mice infected with the S strain virulent die from pneumonia infection, but mice infected with the R strain do not develop pneumonia. Griffith was able to kill bacteria by heating them. He observed that heat killed S bacteria, the bacteria S strain bacteria that he killed by heat and injected into the mice did not kill them. When S strain, normally if we injected into the mice died. But when S strain that was heat killed was injected into the mice lived. When he injected S strain heat killed bacteria injected into the mice will live. But the second thing, the S strain heat killed bacteria plus R strain live bacteria. The R strain is living, but S strain is killed. But this both he mixed and injected into the mice died. He injected a mixture of heat killed S and live R bacteria, the mice still died. Moreover, he discovered living S bacteria from the mice. Basically, S bacteria normally was killed by the mice. I mean, when heat killed S strain bacteria was injected into the mice lived. But when heat killed S strain plus R strain bacteria injected into the mice died. And he also found S strain bacteria inside the mice. What can we assume from this? One, we can assume that somehow due to injecting of this S strain plus R strain, we can assume that the R strain bacteria, due to the presence of S strain bacteria, got transformed into the S strain bacteria, and we know that when S strain bacteria is inside the mice will die. This is the first assumption. The second assumption may be something like the S strain might have survived due to immunity. Let's leave that be. This is the main assumption, that is the R strain bacteria with the presence of the S strain heat killed material got transformed into the S strain bacteria that killed the mice. He concluded that the R strain bacteria had somehow been transformed by heat killed S strain bacteria. Some transforming principle transferred from heat killed S strain had enabled the R strain to synthesize a smooth polysaccharide coat and become virulent. This must be due to transfer of genetic material. However, the biochemical nature of genetic material was not defined from his experiments. Basically, he understood this. He said that some transforming principle transformed R strain to S strain and killed the mice. But he didn't derive what this transforming material actually is. Next, that is a biochemical characterization. What molecule it is? Biochemical characterization of the transforming principle. Prior to work of Oswald Avery, Colin MacLeod, and Macklin McCarthy, which is 1933 to 44, the genetic material was thought to be a protein. Before these people's works, they thought the genetic material was a protein. They worked to determine the biochemical nature of transforming principle in Griffith's experiment. Griffith's experiment was basically this one. Frederick Griffith, the heat killed S and the plus R strain transforming principle. The purified biochemicals, proteins, DNA, RNA, etc., from the heat killed S strain to see which one could transform the live R strain into S cells. Basically, there is the heat killed bacteria, S strain, heat killed. From this remnants of this S strain, they extracted protein, they extracted RNA, they extracted DNA and others. They discovered that DNA alone from the S bacteria could cause bacteria to become transformed. Basically, they did experiment, they purified, and I think they injected the DNA separately, RNA separately, and protein separately into the mice. Into the mice along with R strain. Maybe due to that, where only we know that DNA is the main genetic material. When we inject purified protein from the S strain along with the R strain, there is no change. When we inject RNA plus S strain, there is no change. But we inject DNA, which is the main transforming principle we know, as we are from the future. This DNA will transform the R strain bacteria into S strain bacteria. Seeing this, they thought DNA is a genetic material. And they also discovered that protein digesting enzymes, which is proteases, and RNA digesting enzymes, RNases, did not affect the transformation. They added proteases and RNases to the transformation or the S strain heat killed bacteria along with the R strain bacteria. But when they saw that the mice still died when they added proteases or RNases, but when they added DNases, which can digest DNA, the mice did not die, possibly. So, transforming substance was not protein or RNA. Digestion with a DNA digesting enzyme, that did stop the transformation, suggesting that DNA caused the transformation. They concluded that DNA is hereditary material, but not all biologists were convinced. They are saying that by doing this experiment, they thought that DNA is the hereditary material, but not all the biologists were convinced. Now, the genetic material is DNA, finally completely proving that DNA is a genetic material. Then the unequivocal proof that DNA is a genetic material came from the experiment of Alfred Hershey and Martha Chase, called the Hershey-Chase experiment. They worked with viruses that infect bacteria called bacteriophages. The bacteriophage attaches to the bacteria and its genetic material then enters the bacteria. Basically, bacteriophages are viruses that affect bacteria. And this bacteriophage attaches to the bacteria, attaches, this is the bacteria cell, this is a bacteria, uh, a virus, a bacteriophage will come and attach here, and the bacterial cell and its genetic material then enters the bacterial cell. Bacterial cell treats the viral genetic material as if it was its own and subsequently manufacture more virus particles. Basically, the bacterial cell enters the cell, but the cell, thinking that the viral genetic material was its own genetic material, manufactures more virus particles. Hershey and Chase worked to discover whether it was protein or DNA from the viruses that entered the bacteria. Basically, DNA is the bacteriophage mostly have DNA itself as the genetic material. And so when this DNA's the bacteriophage's genetic material enters the cell, it will get divided. This is a unknown fact. So they wanted to know what is the one, if it is protein or DNA which enters the bacteria and actually is the genetic material which is getting divided here. Genetic material is the one that gets divided. They grew some viruses on a medium that contained radioactive phosphorus and some others on a medium that contained radioactive sulfur. Basically, sulfur is present inside the protein, whereas phosphorus is present inside the DNA, because we know nucleotide is made of phosphate, pentose and nitrogenous bases. So phosphorus is of course present in the inside the DNA, and sulfur is present not in the DNA but only on the protein. Viruses grow in presence of radioactive phosphorus contain radioactive DNA but not radioactive protein, because DNA contains phosphorus but protein does not. Basically, protein doesn't contain phosphorus, so only DNA will become radioactive. Similarly, viruses grown on radioactive sulfur contain radioactive protein but not radioactive DNA, because DNA does not contain sulfur. Radioactive bacteriophages were allowed to attack the E. coli bacteria. Then, as the infection proceeded, the viral coats were removed from the bacteria by agitating them in a blender. Basically, as the infection proceeded, the viral coats, the coats of the viruses, were removed from the bacteria by agitating them in a blender. The virus particles were separated from the bacteria by spinning them in a centrifuge. They centrifuge the virus and bacteria particles after the infection. Basically, after letting the bacteriophage supply its genetic material into the cell. Bacteria which was infected with viruses that had radioactive DNA were radioactive. Bacteria, because DNA is the one that is genetic material, DNA is the one, radioactive DNA from the bacteriophage is the one that goes inside the bacteria. So only bacteria infected with viruses had a radioactive DNA, indicating that DNA was the material that was passed from the virus to the bacteria. Bacteria that were infected with viruses that had radioactive proteins were not radioactive. Bacteria were not radioactive. The proteins were radioactive, but bacteria did not gain anything from the proteins. Basically, after infection or injection from the genetic material, the protein coats will not enter the bacteria but will still be left outside itself. So the outside itself, they separated it, so it will not be present inside the cell. This indicates that protein did not enter the bacteria from the virus. DNA is therefore the genetic material that is passed from the virus to the bacteria. radioactive 335 sulfur labeled protein capsule. Red color is the radioactive one. Here it is DNA, here it is the protein. First stage is the infection where the DNA will enter the bacteria. Here if we see, the protein coats are outside due to blending separation, and inside there is only DNA. DNA is not radioactive, so there is no need for bacteria to be radioactive. Radioactive sulfur 35 detected in the supernatant. No radioactive detected in the supernatant, which is the protein. So here if we see, the DNA itself was radioactive here because phosphorus is in the DNA. So in such a case, the cell also had DNA. This is a Hershey-Chase experiment. Properties of genetic material, DNA versus RNA. From the foregoing discussion, it is clear that debate between proteins versus DNA as a genetic material was unequivocally resolved from the Hershey-Chase experiment. Hershey-Chase experiment basically proved that DNA is the genetic material. It became established fact that it is DNA that acts as a genetic material. However, it subsequently became clear that in some viruses, RNA is the genetic material. For example, tobacco mosaic virus, QB bacteriophage, etc. Basically, we know it became a fact that DNA is a blueprint, DNA is the genetic material. But we also know in some viruses, RNA is the genetic material. Answer to some of the questions such as why DNA is the main dominant genetic material whereas DNA performs dynamic functions of messengers and adapter has to be found from the differences between chemical structures of the two nucleic acid molecules. Why DNA is the main genetic material but DNA is performing dynamic functions as a messenger and an adapter? This can be found in the differences between chemical structure of the two nucleic acid molecules. Can you recall the chemical differences between DNA and RNA? Basically, RNA has an extra oxygen, an extra hydroxy group, whereas there is a lack of oxygen in the hydrogen in case of DNA, deoxy. A molecule that can act as a genetic material, this is the ideal genetic material criteria. It should generate its replication should be there, should be able to generate its replica. It should be able to be chemically and structurally stable. It should be stable chemically and structurally. There should be no strain. It should provide the scope for slow changes, mutation that are required for evolution. If there is no mutation, there is no evolution itself. Because of such a reason, it should be stable, but it should not be completely so stable that there are no changes itself, no matter how much time passes. If such was the case, there used to be no evolution. It should provide scope for slow changes, that is mutation, that are required for evolution. And it should be able to express itself in the form of Mendelian characters. If one examines each requirements one by one, because the rule of base pairing and complementarity, both the nucleic acids, DNA and RNA have the ability to direct their duplication. Basically, both RNA and DNA can undergo replication. They can undergo replication. Both RNA and DNA satisfy the first condition. Let's see the second condition. The other molecules in living systems such as protein fail to fulfill the first criteria itself, but protein can't undergo self-replication. It cannot fulfill the first criteria itself, so it cannot be a genetic material. The genetic material should be stable enough to not change with different stages of life cycle, age, or with change in physiology of the organism. Basically, the ideal genetic material should change every a little bit as the time goes by. That is to allow evolution, but it shouldn't be so unstable that in 100 years itself, if for a human life cycle is 100 years, if RNA is a genetic material, it should not change like every 10 years. It should not change, and every 10 years there should not be a rapid change in the in person organisms physiological features that would lead to very very dangerous issues. It should be stable, at least that, and in the lifetime of an organism it should not change with age, life cycle, etc., or with change in physiology of the organism. Like if a person loses his hand, like that is a change in physiology, but just because of that it should not change. Stability as one of the properties of genetic material was very evident in Griffith's transforming principle itself, that heat, which killed the bacteria, at least did not destroy some of the properties of genetic material. Basically they are saying stability as one of the properties of genetic material was very evident. Basically we saw when we heat killed the bacteria did die, but the protein, I mean not protein, the genetic material inside the bacteria, that did not change, because the next R bacteria, which were influenced by the transforming material, transformed into the same as strain bacteria. There was no change. So we can say that DNA is stable. This is now can be easily explained in the light of DNA, that the two strands being complementary, if separated by DNA, come together when appropriate conditions are provided. Further, 2' OH groups present at every nucleotide in RNA is a reactive group and makes RNA labile and easily degradable. Basically, we can easily explain that DNA is stable and the two strands being complementary, separated by heating, come together when appropriate conditions are provided. Further, 2 OH group present at every nucleotide in RNA is a reactive group, make RNA labile and easily degradable. Because RNA has 2 OH group, it can easily react. It is very reactive compared to DNA, so it is easily degradable. RNA is known to be catalytic, now known to be catalytic. Hence reactive. Therefore DNA chemically is less reactive and structurally more stable when compared to RNA. Therefore, among the two nucleic acid, the DNA is a better genetic material. In the second criteria, where stability comes, RNA fails there. As for the third criteria, if we see, provides scope for slow changes that are required for mutation. RNA does provide change, but it is not slow changes. It is actually a little fast changes. So it might even change very much. So it is not slow changes, and the evolution will be very rapid, and it might lead to a disaster. And the fourth criteria, it should be able to express itself in the form of Mendelian characters. This might or might not be followed by RNA. In fact, the presence of thymine at the place of uracil also confers additional stability to DNA. Place of uracil, there is thymine. This also helps maintain stability of DNA. Detailed discussion about this requires understanding of the process of repair in DNA. You will study these processes in higher class, how this thymine provides higher stability. If you want to understand that, you need understanding of the process of repair in DNA. That you will not be studying right now, but in higher classes. You will not be studying right now, but in higher classes. Both DNA and RNA are able to mutate. In fact, RNA being unstable, mutate at a faster rate. Consequently, viruses having RNA genome and having a shorter lifespan mutate and evolve very fast. RNA can directly code for synthesis of proteins, hence can easily express the characters Basically, for DNA transcription, a process should happen where DNA transforms into mRNA, and mRNA then transforms or produces proteins. But in case of RNA, where RNA is a genome, RNA can easily express the protein. Basically, RNA is the one that will provide the protein, so it can more easily express the physiological characters. The DNA, however, is dependent on RNA for synthesis of proteins. The protein synthesizing machinery has evolved around RNA. The above discussion indicate both RNA and DNA can function as genetic material, but DNA being more stable is preferred for storage genetic material. For the transmission of genetic material information, RNA is better because RNA can easily be formed and it can act as a easy transformation agent. James Watson and Crick model for James Watson and Francis Crick model for semi-conservative replication of DNA. We will see this in the next topic, but before that there is a small topic of the RNA world. From foregoing discussion, an immediate question becomes evident. Which is the first genetic material? What could be the first genetic material? It shall be discussed in detail in chapter on chemical evolution, but briefly we shall highlight some of the facts and points. DNA was the first genetic material. There is now enough evidence to suggest that essential life processes such as metabolism, translation, splicing, etc. evolved around DNA. RNA. Basically, RNA is the first genetic material. There is now evidence to suggest that essential life processes such as metabolism, translation, forming of proteins, splicing, etc. evolved around RNA. RNA used to act as a genetic material as well as catalyst. There are some important biochemical reactions in living systems that are catalyzed by RNA catalyst and not by protein enzyme. But RNA being a catalyst was reactive and hence unstable. Therefore, DNA has evolved from RNA with chemical modification that make it much more stable. DNA being double-stranded and having complementary strand further resist changes by evolving process of repair. Basically, RNA world is a short discussion about what is the first genetic material and how DNA originated from RNA, and how DNA is more stable than RNA because of the complementary structures and pairing and being double-stranded. And they say that there is evidence right now that metabolism, translation, splicing evolved around DNA. Now let's go to the next main topic, that is replication. While proposing the double helical structure for DNA, Watson and Crick had immediately proposed a scheme for replication of DNA. Basically, Watson and Crick proposed many things. The first one was the double helix model of DNA from diffracted X-ray data provided from Morris Wilkins and Rosalind Franklin, I think. And that is the double helix model from seeing the diffraction data, X-ray diffraction data. And this is due to the double helix data is mainly based on two ideas. The first is that the DNA, there are, based on the Erwin Chargaff proposed that the number of purines and pyrimidines will always be same. So based on that, and because of the possible base pairing between the purines and the pyrimidines, based on these two things, they concluded that DNA is a double helical model. And they gave also the central dogma of genetics. Central dogma of genetics. central dogma is a central dogma of molecular biology, which is basically DNA self-replication, transcription gives mRNA, mRNA gives proteins through translation. And they also proposed the replication. Watson and Crick had immediately proposed a scheme for replication of DNA. To quote their original statement, this is as follows. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Watson and Crick, 1953. The scheme suggested that the two strands would separate and act as a template for synthesis for new complementary strands. Basically, the two strands will get separated. This is the structure. This will get separated to this, and each will act as a template to form new DNA. After the completion of a replication, each DNA molecule would have one parental and one newly synthesized strand. This scheme was termed as semi-conservative DNA replication. Basically, when one DNA which has two strands replicates, it will give two DNA with two daughter cells. Basically, it's like a binary fission where a single cell divides to give two cells. Here a single DNA divides to give two DNA. This is what we call a semi-conservative conservation. The experimental proof for this semi-conservative nature, it is now proven that DNA replicates semi-conservatively. It is shown first in Escherichia coli and subsequently in higher organisms such as plants and human cells. Matthew Meselson and Franklin Stahl performed the following experiment in 1958. The statement that the DNA is semi-conservative was given by Watson and Crick. That is true, but the experimental proof was not done by them, but was done by Matthew Meselson and and Franklin Stahl performed the following experiment in 1958. In 1958, they grew E. coli in a medium containing isotope of nitrogen, which is 15 nitrogen ammonium chloride. 15 nitrogen is heavy isotope of nitrogen as the only nitrogen source for many generations. The result was that 15 nitrogen was incorporated into newly synthesized DNA as well as other nitrogen containing compounds. This heavy DNA molecule could be distinguished from the normal DNA by centrifugation in cesium chloride density gradient. Please note that the 15 isotope is not radioactive isotope and it can be separated from 14 nitrogen only based on densities. Basically, they are saying that E. coli, they took E. coli bacteria. E. coli is from here they gave E. coli was used in two experiments. One is the transforming principle experiment by Friedrich Griffith, and now in 1958 in the semi-conservative nature of DNA, which is basically they took E. coli, they took it in a ammonium chloride which is having isotope of a heavier isotope N15 isotope of nitrogen. So basically when they have only that nitrogen from the surrounding, the bacteria when replicating, the DNA will incorporate this nitrogen 15 isotope itself into the DNA. In such a case, during such cases, the DNA, if many generations, if it is left there, the new generations of bacteria will have heavy isotope of nitrogen in their DNA. This is what they're saying. And when there is heavier isotope and lighter isotope, when we centrifugate them, The lighter isotope will be on the top because of the lower density. Density based on separation based on density because of due to gravitational force. Based on densities, mass by volume, it will be separated, due to which we can identify which is the heavier and which is the lighter isotope of the DNA. And this is taken in a cesium chloride solution. That is based on density gradient. And they are saying the heavier isotope of nitrogen is not radioactive. If it was radioactive, there were other methods for finding out if the DNA was having nitrogen or not. But here, this 15N nitrogen is not an isotope. I mean, it's not radioactive isotope. It's only an isotope. Then, after this growing the E. coli in the heavier isotope medium, they transferred the cells into a medium with normal NH4Cl and took samples of various time intervals as a cell multiplied. Basically, first for many days or many generations they kept in N15 medium surrounding many days, and after those many days, one day they took this bacteria out of this N15 medium and they kept this bacteria in a N14 medium. Then when they are dividing, they lost the N15 source. They only have the N14 source. Now the new generations will have N14 incorporated into their DNA. Now let's observe how it will be incorporated. Now they transferred the cells into medium with normal isotope of nitrogen, ammonium chloride, and took samples at various definite time intervals as the cells multiplied, and extracted the DNA that remained as a double-stranded helix. Basically, where in E. coli we see the cycle where each approximately 20 minutes the cells will divide. At these time intervals, the cells multiplied, they extracted the DNA as double-stranded helices. The various samples were separated independently on cesium chloride gradients to measure the densities of DNA. Results are shown in the figure. Basically, if we see in the normal heavy isotope, if we see, in case of, like, if only heavier isotope is there, only heavier isotope will we see at the bottom of the test tube. But in case of here, if we see, we know that semi-conservative how each helix will get separated into two individual helixes, I mean not helix, two individual strands, and each strand will develop into its own DNA. This is what's happening. The new strand that is going to form will be N14 because it is only formed from the N14 itself. And in here, if we see, the new hybrid is having N14 and N15 will be a little higher than the N15 isotopes in case of the position in the test tube, because it is not as heavy as the actual heavy isotope, not light as the actual light isotope. Because of that, it's in the intermediate. The DNA is containing both the individual strands, both the N14 and 15. But in case of the second cycle, after 40 minutes, if we see, with a 20 minutes gap, here this DNA which is containing this hybrid is going to separate into individual strands again like this, and again derive its nitrogen base which is from the surrounding, and this will again transform into a light DNA, a hybrid DNA, and a light DNA again. So in this case we see a separate light solution and a hybrid solution, hybrid position. Thus, the DNA that was extracted from the culture one generation after the transfer from N15 to N14 medium, that is after 20 minutes, E. coli divides in 20 minutes, had a hybrid or intermediate density. DNA extracted from the culture after another generation, that is after 40 minutes, this is from the start or 20 minutes after the first cycle, the second generation was composed of equal amounts of this hybrid DNA and of light DNA. If E. coli was allowed to go for 80 minutes, then what would be the proportions of light hybrid densities of DNA molecules? If it was grown for 80 minutes, then it is in the fourth cycle, where basically in the first cycle it is in the first 20 minutes it was a hybrid, all were hybrids. In the next 20 minutes it became equal proportion, with two being hybrids, two being normal light isotope. In the third cycle, two will remain as hybrids and four will become light isotopes. And in the fourth cycle, which is the 80-minute mark, two will still remain as hybrid and 8 will become light isotope. So the ratio is 8:1 after 80 minutes. Eight is to 1. Wait, not 8:1, it is 8:2 or 4:1. Very similar experiments involving use of radioactive thymidine to detect distribution of a newly synthesized DNA in chromosomes was performed by Vicia faba beans by Taylor and colleagues in 1958. The experiment, not by Vicia faba, it was on Vicia faba, which are basically faba beans. This was performed by Taylor and colleagues, his colleagues, in 1958, basically using radioactive thymidine. The experiments prove that DNA in chromosome also replicates semi-conservatively. The machinery and enzyme for replication. In living cells such as E. coli, the process of replication requires a set of catalyst enzymes The main enzyme is referred to as the DNA-dependent DNA polymerase. One of the main enzymes required for the replication is called as DNA polymerase, since it is used as a DNA template. DNA is used as a template to catalyze the polymerization of deoxynucleotides. Basically, there is a difference here in case of transcription and self-replication. In transcription, only one DNA strand acts as a template, but in case of self-replication or self-conservation, both the strand act as a template. Okay, let's continue. Since it uses DNA template to catalyze the polymerization of deoxynucleotides, basically individual deoxynucleotides will get polymerized in case of the formation of new DNA, and DNA polymerase is used here. These enzymes are efficient enzymes as they have to catalyze polymerization of large number of nucleotides in a very short time. E. coli has around, not only, but 4.6 into 10 power 6 billion base pairs, not billion, basically 4.6 million base pairs, and in compared to human, whose diploid content is 6.6 billion base pairs, completes the process of replication within 18 minutes. That means for DNA polymerase, the average rate of polymerization has to be approximately 2000 base pairs per second. DNA polymerase has to produce 2000 base pairs per second. Each nucleosome has around 200 base pairs, around a single octamer. So in case of this, 10 nucleosomes will be produced per second. That is, if it needs to produce all of them in under 18 minutes. This is basically obtained by giving the number of base pairs by the time 18 minutes. 18 into 60 if you do, will get in terms of seconds. Not only do these polymerizers have to be fast, but they also have to catalyze the reaction with high degree of accuracy. Any mistake during replication would result into mutation. Furthermore, energetically replication is a very expensive process. Basically, they have to be not only fast, but they should also be very, very accurate. And any misaccurate or mistake in the replication can lead to mutation. And this is also this mutation, this replication is also very, very expensive energy wise. Nucleoside triphosphate serve dual purposes. Basically, deoxyribonucleoside triphosphate is a molecule basically, and it serves two purposes. In addition to acting as a substrate, they provide energy for polymerization reaction. Basically, they act as a substrate and they also provide energy for the polymerization reaction. Two terminal phosphates in deoxynucleoside triphosphate. Deoxynucleoside triphosphate. Okay. Terminal phosphates. Okay. Basically, these are high energy phosphates. That is same as the case of ATP. Present the two deoxynucleoside phosphates, triphosphates. In addition to DNA-dependent DNA polymerases, many additional enzymes are required to complete the process of replication with high degree of accuracy. For long DNA molecules, since its two strands of DNA cannot be separated in its entire length due to very high energy requirement, basically so many hydrogen bonds we should separate. Because of that, the energy requirement is astronomical. The replication occurs with small opening of DNA helix, referred to as a replication fork. The DNA dependent, basically when we want to replicate a DNA, when there is a DNA of like this, DNA, if it is 2 meter long, 2.2 meter long DNA, it is not all the 2.2 meter DNA is separated into two separate strands, but it creates a replication fork like this. This is done through an enzyme called helicase, which is a small opening creating one. The DNA dependent DNA polymerases catalyze polymerization only in one direction, that is from phosphate to hydroxy group, 5' to 3'. Basically, it is required to remember that the DNA dependent DNA polymerases catalyze polymerization, that is only in a specific one unidirectional, that is from phosphate to hydroxy group, that is from 5' to 3'. This creates some additional complications at the replication fork. Consequently, one strand, the template with polarity 3' to 5', but the other strand, the replication is basically, if we see here, this DNA polymerase only works from 3' to 5'. It only works from 3' to 5'. But here it is reverse of here. It needs to go from 5' to 3'. Basically, the enzyme will be here. The DNA polymerase will be DNA dependent DNA polymerase will be somewhere here, and it will be on the DNA. It will go like this. That is, if the template strand is from 3' to 5'. But what about in case of the 5' to 3'? It doesn't happen this way. Let's see what they are saying here. Complicating some this DNA dependent DNA polymerases catalyze polymerization only in one direction, that is 5' to 3'. This creates some additional complication at the replication fork. Consequently, on one strand, the template with polarity 3' to 5', the replication is continuous. is 3 prime to 5 prime. Wait. 3 prime to 5 prime. It catalyzes 5 prime to 3 prime. That is a new strand. From 3 prime to 5 prime, it will create a 5 prime to 3 prime strand, so it will be continuous. While on the other hand, the template with phosphate to hydroxy group 5 prime to 3 prime, it is discontinuous because the strand formed should be anti-parallel, so it should be 3 prime to 5 prime. That is the new strand that is going to form. That is from hydroxy to phosphate groups. This, because it only catalyzes reaction from a new strand being 5 prime to 3 prime, phosphate to hydroxy. In such a case, the new strand is discontinuous. The discontinuously synthesized fragments are later joined by the enzyme DNA ligase. Basically, since it only catalyzes from 5 prime to 3 prime, basically it will come here. The enzyme will come here and do this much, again do this much, like this. And these individual fragments are again joined by DNA ligase. We can't leave them as discontinuous fragments, so it is joined by DNA ligase. The DNA polymerases on their own cannot initiate the process of replication. The DNA polymerases on their own cannot initiate the process of replication. Okay, the DNA polymerases are these DNA dependent DNA polymerases enzyme cannot initiate the process of replication on their own. Also, the replication does not initiate randomly at any place in DNA. There is a definite region in E. coli DNA where the replication originates. Such regions are termed as origin of replication. Basically, if there is a strand of DNA, we can't start replication from here. If you want, we cannot start. In here, if you want, we can't start. Or any place like this, we cannot start the replication. There is a specific site for replication that is called origin of replication. Or origin of replication. Origin of at this site only the replication will start. It is because of the requirement of the origin of replication that a piece of DNA, if needed to be propagated during recombinant DNA procedures, requires a vector. The vector provides the origin site of replication. Basically, we cannot start replication at any sites. There needs to be origin of replication. That is why when we introduce like hybrids, when we create new genes, when we introduce new genes into an organism and we want it to replicate, we can't directly introduce into a DNA because there is no site of replication itself. Because of such a reason, we introduce vectors. Vectors are basically some molecule or substances which provide the origin of site of replication. Origin site for replication. This site will allow the replication of the DNA. This we will learn later in biotechnology. Further, not every detail of replication is understood well. Not every detail, even now, even in the modern era, we don't know every detail of the replication very well. In eukaryotes, the replication of the DNA takes place at S phase of the cell cycle. The replication of DNA and cell division cycle should be highly coordinated. A failure in cell division after DNA replication results in polyploidy, a chromosomal anomaly. You will learn the detailed nature of origin and the process occurring at this site in higher classes. And more about this topic is also discussed in the biotechnology chapter. This is the replicating fork. Now let's see one of the main topics, the next main topic, transcription. The process of copying genetic information from one strand of the DNA into RNA is termed as transcription. Basically, copying information from one strand, the template strand of DNA, into RNA is termed as transcription. Here also the principle of complementary governs the process of transcription. Here also the principle of complementarity governs the process of transcription. Except the adenosine complements now form base pairs with uracil instead of thymine. Basically, adenosine nitrogenous base bonds with thymine, but in case of RNA it will bond with uracil. However, unlike in the process of replication, which once set the total DNA of an organism gets duplicated, in transcription only a segment of DNA and only one of the strands is copied into the DNA. Limitation here in transcription is, in DNA all the strand gets duplicated to form double the DNA. All the 2 meter gets separated to form double the DNA, but in case of transcription that is not the case. On the whole DNA only a certain specific part of the segment of the DNA is replicated and only one strand is replicated. The mRNA is single stranded. It's not a double stranded helix or anything like that. It's only a single stranded. mRNA is in this case single stranded. In transcription only a segment of DNA and only one of the strands is copied. This necessitates defining the boundaries that would demarcate the region and the strand of DNA that would be transcribed. Basically, because of this segment, only one segment getting transcribed, we need to define which is the boundary that would transcribe, which part of the DNA actually transcribe, which doesn't, and there is a necessity to define such regions. Why both the strands are not copied during transcription has a simple answer. Before one thing, we saw that euchromatin was nucleosomes arranged loosely. There it could allow transcription, it is active, but in case of heterochromatin, which is tightly bounded together and has a darker strain, takes a darker strain, it doesn't undergo transcription. Okay, I'm reminding something. Okay, now let's continue. Why both the strands are not copied? We saw that only one strand is copied here. Why both the strands are not copied during transcription? The simple answer. First, if both strands act as a template, they would code for RNA molecules with different sequences. Remember, complementarity does not mean identical. And in turn, if they code for proteins, the sequence of amino acids in the proteins would be different. Basically, they are saying if both strands act as a template, like in semi-conservation of DNA, replication of DNA, they could code for RNA molecule with different sequences. Yes, they will be exactly opposite sequences to each other, but exactly same sequences as the DNA, except in place of thymine there will be uracil. And because of the different sequences, the protein they would code for also will be different. Hence, one segment of the DNA would be coding for two different proteins. One segment of the DNA, each segment of the DNA will be coding for one segment of the double helical DNA, not a strand. One segment is going to code for two different proteins, and this would complicate the genetic information transfer machinery. This will be very complicating the thing, so it's not done. Second, the two RNA molecules if produced simultaneously would complementary to each other, hence would form a double-stranded RNA. Because they are complementary, they will stop doing translation and they will form a complementary RNA. This would prevent RNA from being translated into protein and exercise the need for transcription. And this transcription will become, when there is a double-stranded RNA, there is no translation. If there is no translation, there is no use for transcription only, or become futile. Transcription unit. A transcription unit in DNA is defined primarily by three regions in the DNA. First one, a promoter, a structural gene, a terminator. Promoter starter, terminator ender. there is a convention in defining the two strands of DNA in the structural gene of a transcription unit. There is a convention in defining the two strands of DNA in the structural gene of a transcriptional unit. Since the two strands have opposite polarity and the DNA dependent RNA polymerase also catalyze the polymerization in only one direction, that is 5' to 3', the strand that has a polarity 3' to 5' acts as a polymerase and referred to as a template strand. Basically, there is a convention in defining two strands of the DNA in the structural gene of transcription unit. Since two strands have opposite polarity, they are saying there is actually a convention, basically a traditional way in defining the two strands of the DNA in the structure of a transcription unit. A traditional way, that is, since the two strands are antipolar, meaning different polarity or opposite polarity, and then a DNA dependent polymerase also catalyzed polymerization only in one direction, that is from 5' to 3'. We saw in DNA polymerase, DNA dependent DNA polymerase, it also catalyzes a reaction from 5' to 3'. That is the strand that is going to get coded for. The template should be from 3' to 5'. The coded strand or the newly produced strand should be from 5' to 3', phosphate to hydroxy. But the template strand should be from 3' to 5'. DNA dependent RNA polymerase also does the same thing. It also catalyzes reaction only in one direction, that is the same as the DNA dependent polymerase, that is also from 5' to 3'. The newly produced strand should have 5' to 3' polarity and the template strand should have a polarity of 3' to 5'. This is also referred to as a template strand, that is 3' to 5'. The other strand, which has a polarity 5' to 3' and a sequence same as RNA, except thymine at the place of uracil is displaced during transcription. It is moved away from the transcription unit. Strangely, this strand, which does not code for anything, is referred to as a coding strand. It's not acting as a template, but it's called as a coding strand. Maybe it is because it has the same sequence as a coded strand, except in place of uracil, in place of thymine, there is uracil in the coded strand. All the reference point while defining a transcription unit is made with a coding strand. All the reference point while defining a transcription unit is made with coding strand. The reference point is basically the coding strand. To explain the point, a hypothetical sequence from a transcription unit is represented below. It's not a real sequence, it's just represented here. Because a DNA dependent RNA polymerase acts only in one direction, that is 5' to 3', the template strand can only be from 3' to 5'. So this will be the template strand. This ATGCA are basically adenosine, thymine, guanine and cytosine. Okay, this is about template and coding strands. This is a schematic structure of a transcription unit containing a promoter, structural gene, and a terminator. The coding will, the DNA dependent RNA polymerase will code from 5 prime to 3 prime, and so the template strand should be from 3 prime to 5 prime always. The promoter and terminator flank the structural gene in a transcription unit. The promoter is said to be located towards 5 prime end, which is basically the phosphate end, which is upstream of the structural gene. The reference is made with respect to the polarity of the coding strand. Basically, from 5 prime to 3 prime is called an upstream strand. I think it is a DNA sequence provides binding site for the RNA polymerase, and it is the presence of a promoter in a transcription unit. It also defines the template and coding strands. It is a DNA sequence that provides the binding site for the RNA polymerase, the DNA sequence itself, where the polymerase will attach itself, and it is the presence of a promoter. It is the presence of a promoter in transcription unit that also defines the template and coding strand. This is a statement-based question. This could be asked for a statement-based question. The presence of a promoter and transcription unit that also defines the template and coding strands. By switching its position and with terminator, the definition of coding and template strands could be reversed. Okay. The terminator is located towards the 3 prime end downstream. 5 prime is the upstream and 3 prime is the downstream of the coding strand, and it usually defines the end of the process of transcription as it happens from 5 prime to 3 prime. 5 prime to 3 prime is the coding. So 3 prime is the coding strand, usually defines the end of the DNA will be produced this side, where this will be the 3 prime and this will be the 5 prime. Terminator here is present at the, because it's a convention, we define everything with reference point of the coding strand. That is why maybe 5 prime is upstream and 3 prime is the downstream. There are additionally regularly sequences that may be present further upstream or downstream to the promoter. Some of the properties of these sequences shall be discussed while dealing with regulation of gene expression. Transcription unit and the gene. A gene is defined as a functional unit of inheritance. Though there is no ambiguity that the genes are located on the DNA, it is difficult to literally define a gene in terms of DNA sequence. Basically, a gene is defined as a functional unit of inheritance. Though there is no ambiguity that we can say that genes are located on the DNA. We can indeed say genes are located on the DNA, but we cannot literally define a gene in terms of a DNA sequence. Like this much segment of a this much DNA segment is called a single gene, or this much amount of base pairs or DNA segment is called the 2 genes or something like that. We cannot define it like that. It is very difficult to do so. The DNA sequence coding for tRNA and rRNA molecules are also define a gene. However, by defining a cistron as a segment of DNA coding for a polypeptide, the structural gene in a transcription unit could be said as a monocistronic or polycistronic, mostly in bacteria or prokaryotes. Basically, we are defining a gene. The DNA sequence coding for, we can define the DNA sequence coding for tRNA and rRNA molecules, which is transfer RNA and ribosomal RNA are molecules also has a gene. However, by defining a cistron as a segment of DNA coding for a polypeptide. Polypeptide means a protein. Cistron is the amount of segment, maybe 2000 base pairs, 3000 base pairs, 20,000 base pairs. That specific amount of base pairs or that specific amount of the segment coding for a specific polypeptide, it is called a cistron. We are defining cistron in terms of a segment of a DNA. This is not a strand of a DNA, a segment of a DNA. Polypeptide being a protein. The structural gene could be said as the structural gene in a transcription unit could be said as a monocistronic. This is mostly in eukaryotes, or polycistronic, mostly in bacteria or prokaryotes. In eukaryotes, the monocistronic structural genes have interrupted coding sequences. The genes in eukaryotes are split. The coding sequences or express sequences are defined as exons. Exons are said to be those sequences that appear in mature or processed DNA. The exons are interrupted by introns. Introns are intervening sequences that do not appear in mature or processed DNA. The split gene arrangement further complicates the definition of a gene in terms of a DNA segment. Basically, we cannot define a gene as this much amount of a segment, which is very hard to do. So we define, instead of that, basically a DNA sequence coding for any kind of molecule, tRNA, rRNA, any kind of molecule, it can be defined as a gene. However, we can define another special term as a cistron. A cistron is basically a segment of DNA, no matter how much long it is, depending for the specific protein. The cistron is a segment of DNA which codes for a polypeptide, which is coding for something. It is acting as a structural gene in the transcription unit. And this structural gene can be monocistronic, this is mostly in eukaryotes, or polycistronic, mostly in bacteria or prokaryotes. Monocistronic means, I think, coding for a single kind of polypeptide. And polycistronic meaning that segment is coding for many kind of polypeptides. Is monocistronic in DNA mean coding for a single segment, coding for a single protein or polypeptide, and polycistronic mean a single segment of DNA coding for many polypeptides. Single mRNA molecule transcribed from DNA. So it's not DNA that's called monocistronic, but rather mRNA. The DNA segment will have one gene, one mRNA, one protein. Polycistronic, a single mRNA molecule encodes multiple polypeptides, usually part of the same pathway or complex. This is common in prokaryotes, especially in operons like the lac operon, which we will study in the further of this topic. A single promoter devices the transcription of multiple genes. One long mRNA translated into several proteins. isn't defined, no, is histron defined as a length of DNA or as or only as the specific mRNA formed after transcription. Histron is defined as histron is defined at the DNA level, not the mRNA transcript. The terms histron originate from the class, okay. Basically, cistron DNA level, segment of DNA, okay. Cistron is correct. Cistronic is in terms of the mRNA. Cistron is DNA length itself. And the next information they gave, polycistronic is in prokaryotes, and in eukaryotes, the monocistronic structural genes have interrupted coding sequences. Basically, only one part of the, I think that is what it means. Interrupted coding sequences which are exons and introns. Does that mean only exons carry sequence which undergo transcription? Or does that mean the RNA, more specifically mRNA, have both introns and exons, but only the exon sequences undergo translation. Interrupted coding sequences. This refers to genes mostly in eukaryotes. Exons are coding or expressed regions. Introns are non-coding regions in interspace between exons. Both exons and introns are transcribed into the initial mRNA called pre-mRNA or hnRNA. So yes, the mRNA at first has both introns and exons. What happens after transcription? Splicing occurs, all the introns are removed and exons are joined together. This creates the mature mRNA, which is shorter, has only exons, and is now ready for translation. Only exon sequences remain in the mature mRNA and are the ones that are translated into polypeptides. Exons are not the only parts transcribed. Both exons and introns are transcribed, but exons are the only one undergo translation. Then what is the need for the DNA to contain these useless sequences? Now actually before that is exon and intron defined for DNA or they are parts of the gene in DNA, not of mRNA. DNA sequences with a gene that will be retained in the final mature mRNA and may be translated into protein. Introns, DNA sequences with a gene that are transcribed into pre-mRNA but removed during. Okay. Structure of the gene on the DNA. Observe their effects in mRNA, but we define them based on DNA. DNA contains introns, contains exons, yes. Pre-mRNA contains introns, yes. Exons also it contains. It is not defined in the pre-mRNA or mRNA. It is defined, exons and introns are defined only for the DNA. Mature mRNA also doesn't contain introns, doesn't contain exons, and it cannot be defined. So why does DNA even need to have introns, which I won't say useless, but first guess of mine relation to stability of DNA. My second doubt, I don't think it is mostly not first one. It might be yes, but I don't think so. The second one, I think the reason where many other actually I got one more doubt. Is intron and exon defined for a single polypeptide, single mRNA as a reference, or is it constant for all DNA. Exon and intron structure is defined in DNA, so it is constant in the genome for that gene. In specific mRNA can vary due to alternative splicing. So many different mRNAs can be made from the same gene using different combination of exons. So the correct answer is exons and introns are defined in DNA, which show exon shows up in mRNA depends on how pre-mRNA is sliced. Sometimes exons are potential coding sequences. Whatever they end up in the final mRNA depends on splicing. Why do introns exist at all? Scientists doesn't seem to know this, but there are some of the best supported reasons. Alternative slicing, main reason, allows a single gene to give rise to multiple proteins depending on which exons are included. This increases protein diversity without increasing the number of genes. For example, the human body has 20,000 genes but makes over 100,000 different proteins thanks to alternative slicing. Regulatory roles. Introns contain regulatory elements like enhancers or silencers. Affect gene expression. Okay. Role when, where, and how much gene is expressed. Evolutionary flexibility. Introns make it easier for exon to be shuffled or duplicated, allowing the evolution of new proteins via exon shuffling. Protection buffering. Some theories suggest that introns may be protect coding regions from mutations. Since they are non-coding, mutations in introns is less likely to harm the organism. They are not primarily due to DNA stability. Their presence is more about gene regulation, protein diversity, and evolution. Okay. If it says something about 20,000 genes and 100,000 proteins, but if that is the case, isn't that polycystonic? Then it's not found in eukaryotes, right? Genes, 100,000 proteins, isn't that polycystonic? Not found in eukaryotes. Eukaryote genes are still more. Each RNA transcript still codes for. Okay. Okay. So basically, if each RNA transcript is unique, but in case of polycystonic, the mRNA transcript will remain same, but still it can give rise to many proteins. Gives rise to many proteins. Transcript is called, okay, so that is the case here. Fine. let's continue. Let's read the statement again. The DNA sequence coding for tRNA molecules or rRNA molecules is also defined as a gene. However, by defining a cistron, transcription unit could be said by defining a cistron as a segment of DNA coding for a polypeptide, the structural gene in a transcription unit could be said as monocistronic, mostly in eukaryotes, or polycistronic, mostly in bacteria or eukaryotes. I mean prokaryotes. In eukaryotes, the monocistronic structural genes have interrupted coding sequences. The genes in eukaryotes are split. The coding sequences are expressed. Sequences are defined as exons. Exons are said to be those sequences that appear in mature or processed RNA. The exons are interrupted by introns. Introns or intervening sequences do not appear in mature or processed RNA. The split gene arrangement further complicates the definition of a gene in terms of a DNA segment. As we saw before, like 100,000 proteins from 20,000 genes. This is based on due to the exon shifting, or basically exon shuffling that happens. This will change the amino acids in the protein chain, so new proteins will be produced. So it is complicating the actual definition and creating more diversity, more variation, or more uses. Inheritance of a character is also affected by promoter and regulatory sequences on the structural gene. Inheritance of a character is also affected by promoter and regulatory sequences of a structural gene. Hence, sometimes the regulatory sequences are loosely defined as regulatory genes, even though these sequences do not code for any RNA or protein. Basically, inheritance of a character is also affected by promoter and regulatory sequences of a structural gene. And sometimes regulatory sequences can also be defined as regulatory genes. Okay, basically they are saying that inheritance of a specific character, basically this character is produced by proteins or physiologically expressed by proteins. So in such a case, character is also formation of from gene only. It is this gene is actually affected by instead of saying gene here, they said inheritance of character. And also like transfer also we can say is also, this gene is also affected by the promoter and regulatory, which means terminator. We can say it's a terminator, regulator terminator, because regulator is controlling, terminator also is ending the sequence. And transcription unit contains only promoter and terminator with along with structural gene, and hence sometimes the regulatory sequences are loosely defined as regulatory genes because they are also doing a function. We define them as regulatory genes, even though this sequence do not code for RNA protein. Normally we call genes, we call something gene because it is producing a polypeptide chain or a protein or RNA or something like that, but they are not producing, but they are affecting the process. So that is why we call them as regulatory gene. Types of RNA and the process of transcription. In bacteria, there are three major types of RNA: mRNA, messenger RNA, tRNA, transfer RNA, and rRNA, ribosomal RNA. All three RNAs are needed to synthesize a protein in a cell. Basically, there are three types of RNA: messenger, transfer, and ribosomal. All three of these RNA needs to be present to synthesize a protein in a cell. The mRNA provides the template, messenger RNA provides the template which is helping in providing the protein. tRNA brings amino acids and reads the genetic code. tRNA is reading the genetic code through codons which we'll read further in the chapter. And rRNAs play structural and catalytic role during translation. rRNA They did not define it clearly here, but they mainly play. We are going to mostly study in this chapter about rRNA and not rRNA, mRNA and tRNA. mRNA we are studying based on here, which is transcription process. tRNA we are going to study more about in the translation process. And these rRNA play a structural and catalytic role, increase the rate of reaction, or structural role in during translation. There is a single DNA dependent RNA polymerase that catalyzes transcription of all types of RNA in bacteria. Basically, there is a single DNA dependent RNA polymerase that catalyzes transcription of all types of RNA in bacteria. They're saying that there is an DNA dependent RNA polymerase. This is a single enzyme that will catalyze all types of RNA production in bacteria. RNA polymerase binds to promoter and initiates transcription. Initiation, it uses nucleoside triphosphates as a substrate. Basically, they're saying RNA polymerase binds to promoter and initiates the transcription process. It is nucleoside triphosphate as a substrate. We saw this nucleoside triphosphate is also acting as an energy provider for the transcription process itself, even in case of, I think, self-replication. Yes, self-replication, it is acting like similar to these bonds are high energy bonds of the triphosphate bonds, similar to adenosine triphosphate ATP. These nucleoside triphosphates are also having high energy bonds. This also acts as a substrate. This is a process of transcription in case of bacteria, where this is the promoter region, RNA polymerase, sigma factor, DNA helix. Normally the polymerase reaction happens from 5' to 3', which is DNA dependent RNA polymerase happens from 5' to 3'. So the template strand should be 3' to 5'. 3' to 5'. Here they give red and white blue strands here. So the red strand, which is from 3' to 5', is the template strand here. That is why we see it is actually formed from the template strand, which is from 3' to 5'. Here it is the initiation stage. Here it happens in the promoter. It binds the promoter, and this is the elongation phase where RNA is elongated, and the ending phase where the sigma factor has transformed into the rho factor. This is still the RNA polymerase, and polymerizes in a template dependent fashion following the rule of complementarity. It uses nucleoside triphosphate as a substrate and polymerizes in a template dependent fashion following the rules of complementarity. It somehow also facilitates opening of the helix and continuous elongation. Only a short stretch of RNA remains bound to the enzyme. Okay, as we saw here, the entire RNA is outside, but a little RNA is bound to the enzyme. Once the polymerases reach the terminator region, the nascent RNA falls off. So the RNA polymerase, so also the RNA polymerase. Basically, the nascent RNA, we saw that there is a premature RNA which has both the regions, introns and exons. Here they didn't show. Okay, this is I think this might be this is in the bacteria, so prokaryotes, so they didn't show the introns and exons here. Introns and exons here. Only a short stretch of RNA remains bound to the enzyme. Once the polymerases reach the terminator region, the nascent RNA falls off, so also the RNA polymerase. This results in a termination of transcription. An intriguing question is that how is the RNA polymerases able to catalyze all three steps, which are initiation, elongation, and termination. The RNA polymerase is only capable of catalyzing the process of elongation. It associates transiently with initiation factor. Basically, the only factor RNA polymerase, DNA dependent RNA polymerase, can catalyze is the elongation phase, which is the formation of RNA, increase in size of the RNA, basically increasing the number of RNA sequences. That is the only thing it can catalyze. But we saw that RNA polymerase is used in both the promotion, elongation, and termination phases. How that happens is when RNA polymerase only catalyzes the elongation phase, it is that it associates transiently with initiation factor, sigma factor, and termination factor, rho factor. It is associating, it's not actually the one doing the things, it is the initiation factor and termination factor that initiate and terminate the transcription. Association with these factors alter the specificity of the RNA polymerase to either initiate or terminate. Basically, they will show or tell the RNA polymerase whether to promote, initiate, or to end the transcription. In bacteria, since the mRNA does not require any process to become active, and also since transcription and translation takes place in the same compartment, basically in bacteria, since mRNA doesn't need any processing to become active, in mRNA in bacteria doesn't require any processing to become active, it is already mature. And also since transcription and transcription take place in the same compartment, they're saying there is no separation of cytosol and nucleus in bacteria. Many times the translation can begin much before the mRNA is fully transcribed. Basically they're saying basically it will happen both at the same time. Transcription and translation both happen at the same time, and at one side transcription will be going on, at the other side translation will be going on. So it's a one-step kind of process. It is going from one machinery to directly to other machinery without any processing time or any rest time or something like that. It's because there is no separation between the cytosol and the nucleus in the bacteria. Nucleus and cytosol regions are not separated. Consequently, the transcription and translation can be coupled in bacteria. And the next is the transcription in eukaryotes. There are two additional complexities. There are at least 3 RNA polymerases in the nucleus in addition to the RNA polymerase found in organelles. In prokaryotes, we saw there is only a single RNA polymerase, but in case of eukaryotes, there are at least 3 RNA polymerases in the nucleus, and there are also RNA polymerases found in the organelles. So this is more types of RNA polymerases. There is a clear-cut division of labor. Here only one kind of RNA polymerase does one kind of work. It may be initiation, elongation, or termination, or even other works. Only one specific kind of RNA polymerase does that work. That is what we call as division of labor. The RNA polymerase transcribes the RNA polymerases 1 transcribe rRNAs 28S, 18S, 5.8S, whereas I think is a Swedberg unit. In case of we saw 70S and 80S ribosomes in the unit of cell unit of life chapter, or sedimentation unit or coefficient, whereas the RNA polymerase 3 is used for transcription of tRNA, 5sRNA, and snRNAs, small nuclear RNA. We can see the first RNA polymerase 1 transcribes rRNAs, which can be of 28S, 18S, and 5.8s, whereas the RNA polymerase 3 is responsible for transcription of tRNA, transfer RNA, 5s RNA, small nuclear RNAs, and the RNA polymerase 2 transcribes precursor of mRNA, the heterogeneous nuclear RNA, hnRNA. One, like polymerases, the RNA polymerase, DNA dependent polymerase 1 helps in transcription of rRNAs. The RNA polymerase 2 helps in transcription of rRNAs, small nuclear RNAs, tRNAs, srRNAs, 5srRNAs, and the RNA polymerase 2 helps in the transcription of the precursor of mRNA, which is the heterogeneous RNA. This should be remembered. The second complexity is that the primary transcript contains both the exons and the introns, which we saw before. Hence, it is subjected to a process called splicing, where the introns are removed and exons are joined in a defined order. Heterogeneous RNA undergoes additional processing called a capping and tailing. In capping, an unusual nucleotide, methylguanosine triphosphate, is added to the phosphate end of the hnRNA. In tailing, adenylate residues 200 to 300 are added at the 3' end, hydroxy end, in a template independent manner. It is fully processed hnRNA, now called mRNA, that is transferred out of the nucleus for translation. I have a doubt, is this heterogeneous RNA the same as the premature RNA? Yes, okay. Yes, they are the same thing. hnRNA and pre-mRNA are essentially the two things basically. Just to protein coding transcript before they are processed. In practical terms, in broader term, it includes pre-tRNA, unspliced RNA, and some even partially spliced intermediates. But not hnRNA is pre-mRNA. It might be non-coding, still to be translated. Is all hnRNA or mRNA unspliced? All mRNA starts off unspliced, but not all hnRNA is unspliced, and mature mRNA is spliced. hnRNA includes newly transcribed pre-mRNA, completely unspliced, partially spliced intermediates, non-coding RNAs, and other RNA fragments in the nucleus. So, mature mRNA is fully spliced, 5-capped, and so capping is basically adding a nucleoside group to the 5' end and something else to the 3' end of RNA. What is the use? It is 5 prime capping and 3 prime polyadenylation. 5 capping and 3 capping when happens very early during transcription. A modified guanine nucleotide methylguanosine is added to the 5 prime end of the pre-mRNA. It is connected via special 5 prime to 5 prime triophosphate linkage, which is unusual in biology. So yes, it is adding, but it's highly modified. Protects mRNA from degradation by exonucleases, helps ribosomes recognize the mRNA for translation, aids in nuclear export of the mRNA, helps with splicing of the first intron in some cases. Polyadenylation, a poly, a tail about 100 to 250 adenine nucleotides, so many nucleotides, is added to the third end. It is not templated from DNA, it is added enzymatically. Functions of the poly A tail: stabilizes the mRNA, protects from exonucleases, assists with nuclear export, enhances translation efficiency by forming a loop with a 5 prime cap through processes like PABP. Determinants mRNA half-life, longer tails generally mean longer survival. Analogy, think of, okay, fine. Protective plug at the top so it's not corrupted, and the polyadenosine chain, which is around 200 to 350 adenosides, adenosine groups. Adenine are not adenosine or adenotide, it is adenine nucleotides. The second complexity is the primary transcripts. Both contain the exons and the introns and are non-functional. Hence, it is subjected to a process called splicing, whereas the introns are removed and exons are joined in a defined order. hnRNA undergoes additional processing called as capping and tailing. In capping, an unusual nucleotide methylguanosine triphosphate is added to the 5 prime end of the hnRNA, now called mRNA, after the full splicing, that is transported out of the nucleus. In capping, it undergoes additional process called capping and tailing. The capping, an unusual nucleotide methylguanosine triphosphate, is added at the 5 prime end of the hnRNA. In tailing, adenylate residues, basically 200 to 300 adenine nucleotides are added. The significance of such complexities is now beginning to be understood. The split gene arrangement represent probably an ancient feature of the genome. The presence of introns is reminiscent of antiquity, and the process of splicing represents the dominance of the RNA world. In recent times, I didn't get that one, but let's continue for now. The understanding of DNA, RNA and RNA dependent processes in the living system have assumed more importance. Understanding of RNA and RNA dependent processes in the living system have assumed more importance. Okay, let's see the statement. The split gene arrangements represent probably an ancient feature of the genome. The presence of intron is a representative of antiquity, and the process of splicing represents the dominance of the RNA world. The split gene arrangement represent probably an ancient feature of the genome. Split gene reference to the exons coding and introns non-coding. This arrangement where a gene is not continuous but broken into coding and non-coding is likely very old in evolutionary history. It's not a recent invention. It probably existed in early ancestors of the modern organisms. The presence of introns, reminiscent of antiquity, reinforces the idea. Introns may be a leftover feature from a very early stage of life, a genomic puzzle so to speak. Many scientists believe that early life forms had messy, inefficient genes with lots of non-coding junk, and introns are a trace of that time. The process of splicing represents the dominance of RNA world. The RNA world hypothesis suggests that before DNA and proteins evolved, RNA molecules were the main molecules of life, doing both genetic storage and catalysis. Splicing, the process that removes introns from pre-mRNA, is performed by a complex called the spliceosome. The core catalyst activity of splicing is done by RNA, not protein. The core catalytic activity, since RNA acts like an enzyme, it supports the idea that RNA came before proteins. RNA was the main molecule. Okay. Basically, in RNA world, basically everything, I thought it was around only the genetic material, which is RNA, but it seems the RNA world is a suggestion that everything before anything, proteins, any kind of molecules, RNA is the first one to ever exist, or something like that. And yeah, the fact that our genes have introns and require RNA based splicing suggests that these features are very ancient. They are leftovers from a time when life was dominated by RNA, not DNA or proteins, time we call the RNA world. Okay, that's interesting. Ten to 15 pages left. I will continue it in the next class. See you later. Good night.